Hi everybody. Today we're going to talk about the genome organization in bacteria. The term genome, as we imply, we all know that it is a genetic material, and the organization of that genetic material in bacteria is something of much importance because we know the size of the bacteria itself is about zero point 0.2 micrometer in size, the average size. And so into that small size we have to accommodate a large amount of genetic material. And in the case of bacteria, usually it has been found to be a DNA like material. So to fold and pack so much amount of genetic material into the small space of zero point 0.2 micrometer is in fact a big challenge. So let's see how that is going to happen. Now if you go to consider E. coli, the most commonly used organism that we have here, it has a size of about 4.6 million base pairs. Okay, you know what is a base pair, and among that 4.6 million base pairs are there in the E. coli. And in the case of Haemophilus influenza, it has about 1.8 million base pairs. Now if you look at the picture here, you can see that the genetic material in the case of bacteria, they are usually been found to be circular, and they are being they have a usual circular single chromosome which has been present, and since they do not have a true nucleus, this genetic material is being placed within the nucleoid. And because the chromosomes contain only one copy of a gene here, the prokaryotes are considered to be haploid. So if you look deep down into this picture itself, you can see that we do expect that 4.6 million base pairs of E. coli is to be arranged into the small size of about 0.2 micrometer of this E. coli. Similarly, about 1.8 million base pairs have of Haemophilus influenza is also been expected to be arranged in the nucleoid part. Okay, so let's go deep down, but this is already happening, and that's what we are learning about. Now, to imagine this for circular chromosome, just imagine a situation in which you have a Great Wall of China which has been found to be circular. Okay, and this circular chromosome, this Great Wall of China, as I told you, that is of the size, if you extend it, it is of the size of about 350 micrometer. So I told you the average size is about 0.2 micrometer that we have. So if we extend this whole DNA as such, it have a size of about 350 micrometer. Okay, just imagine that at the center of this circle, okay, I'm imagining the DNA, chromosomal DNA, as a Great Wall of China, a circular one. Just imagine there are some people standing in the middle of the circle and aiming at the circle with some arrows so that they can pull a bit of this circle towards the center. Like that, many people are standing and aiming at different points of the circle. In such an imagination, you can find that when they pull the bits of this DNA towards the center, it will result in the formation of various loop domains, and that's which has been depicted in this second picture. And as a result, what happens, the 350 micrometer size of the whole circular chromosomal DNA can be reduced into 30 micrometer. Okay, so from 350 micrometers we have converted into a 30 micrometer with the help of some people who pull the DNA towards the center so that it forms loop domain. And these people who we expect to pull are histone like proteins. In the case of bacteria, of course you cannot have people or anything in the story, but these are histone like proteins which mainly act on the DNA, and they pull the DNA towards the center, forming certain loop domains. And as a result, about 50 chromosomal domains will be formed within a E. coli. Sometimes that will vary, it will vary. So, and at the next stage, what happens? This loop domains will further undergo various supercoilings, and thereby then again the size will decrease. So you do remember, we have to fit it within a 0.2 micrometer size of bacteria. So to enable a macromolecule like DNA to fit within the bacterium, histone-like proteins will bind to the DNA and segregating the DNA molecule into 50 chromosomal domains so that it becomes more compact. And to make it, I told you, to make this happen, there are certain proteins which are there, and further to make it more supercoiled, you need the help of certain proteins such as the DNA topoisomerase enzymes called the DNA gyrase supercoils each domain around itself, forming a compacted mass of DNA which is approximately 0.2 micrometer in diameter. Now, supercoils can be inserted as well as removed by topoisomerases. This is a picture of an electron microgram of a bacterial chromosome which has been to a size of about 1 micrometer. So in an actively growing bacteria, projections of the nucleoid extend into the cytoplasm. Presumably, these projections contain DNA that is being transcribed into mRNA. And the supercoils are both inserted and removed by topoisomerases. So if you look like this is a picture that which you get when an actively dividing cell is being looked. So at some places you can find that the nucleoid is extending and they are being left loose. They are not compacted. So but at some places they are being found to be compacted. The DNA are being found to be compacted also. So the regions where the DNA, the supercoils are being loosened or where projections are happening, those are regions where the DNA is being transcribed into mRNA. Okay, and the regions where they are compacted, it indicates that their RNA synthesis is not happening. So depending upon which all genes has to be transcribed, that all regions of the DNA will be extended. And all these supercoil insertion as well as removal is being done by topoisomerases. And this supercoiled DNA now must be uncoiled and relaxed in order for the DNA polymerases to bind for DNA replication and the RNA polymerase to bind for transcription of the DNA. A topoisomerase called the DNA gyrase catalyzes the negative supercoiling of the circular DNA found in the bacteria, and on the other hand, topoisomerase 4 is involved in the relaxation of supercoiled circular DNA, enabling the separation of the interlinked daughter chromosomes at the end of bacterial DNA replication. So when we have to consider the case of relaxation of the coiling of the supercoils, there are two instances in which this has to be done. One is when the DNA replication has to be done. For that, the DNA polymerase has to come and bind. That is a site where the DNA polymerase has to come and bind. The DNA becomes unbound. The second condition is in which when you need to have to synthesize RNA. In that case, the enzyme RNA polymerase has to come and bind. So in both these cases, the supercoil has to be uncoiled and relaxed, and that has been brought about by the same enzyme called the DNA topoisomerases. So different forms of these topoisomerases will act, one for supercoiling and one for unbinding or the relaxation. In addition to this, as we told earlier, histone-like proteins are also there, which we saw earlier. They also bind to the DNA and aid in the DNA packaging. And other proteins bind to the origin of replication, the location in the chromosome where the DNA. So, the origin of replication is the place where the DNA replication starts. Even there, some proteins will bind. Thank you.